great to be here. Thank you, Albert and John, for inviting me. So today, what I'd like to do is tell you a little bit about the work that we're doing and its impact on how we as a community need to begin thinking about ethical issues related to treating intellectual disabilities, whether they're in Down syndrome and others. This is an area of quite a lot of controversy, particularly because traditionally intellectual disabilities, in particular in Down syndrome, were regarded as disorders that were not treatable and largely not wanted by our society. And as such, there was a move for many, many years until the 60s that children with disability with Down syndrome should be institutionalized, and if possible, and you could detect the disability, or the trisomy early on in children or babies, that they should be aborted. So the studies that we've been doing are actually affecting that discussion, because it argues that, like any psychiatric disorder, that intellectual disabilities can be treated with drugs that actually act on the core issues that are underlying brain dysfunction. So what I would like to do is, I have like 4 topics. So I'm going to try to compact what I want to say in about 30 minutes, and then we're going to talk about some of these issues. So what I'd like to do to give you kind of a concept is, what is Down syndrome? How does it affect the brain? How do these differences in brain abnormalities cause dysfunction? What are just some of the emerging strategies that are coming online that will treat these dysfunctions? And then we'll discuss some of the ethical concerns. Now, to prompt you a little bit in your thinking as I go along, so you can kind of be playing with some of these issues, there are many others. Of course, there's the one related to abortion and whether we should do this. But some of the others that I have listed here are, like, for example, what are the pros and cons of implementing strategies that would improve cognitive function? What factors should parents take into account when making choices for kids that have disabilities, because they can't make those decisions for themselves? Questions of identity. What does it mean to improve cognition in an intellectually disabled person? Should it matter if the drug changes their personality? Interesting question, right? What are the implications of drug therapy for prenatal testing and decisions about termination of pregnancy? An interesting question, of course, is what are the short and long-term consequences of drugs, especially if they're ones that you have to give every day for much of the individual's life. Should we give them if it's going to cause some adverse effect that you can't see, or if you eventually find out that it will shorten the life of the individual by 20 years, should you still do it? Things like that, okay? So those are just something for you to think about. So let's begin by just chatting a little bit about what is Down syndrome, how it's caused, and what are the current strategies that are being used to develop therapies. So I'll tell you a little bit more about this, but it's caused by the triplication of chromosome 21, which has about 200 to 300 genes on it. It's one of the most common disorders, genetic disorders. It affects about 1 in 600 births. Where the incidence is higher in mothers over 35. In the U.S. alone, there are about 350,000 affected individuals, 500 in Europe, and greater than 3 million worldwide. The impairment can be mild to severe, wherein if you measure it by IQ, which is not really very easy to do in someone with an intellectual disability, it can be as low as 20. Which is someone who is largely unable to care for themselves and sit lethargically in a corner. To individuals in their 80s and 90s that are actually able to function quite well. They're considered high-functioning individuals with Down syndrome, and they often have jobs and live and work in our societies. Now, the cognitive impairment begins already within the first year of life, and it more than a decline represents a lack of improvement over time, whereas you know, a normal child will have just kind of exponential growth in their capacity to learn and you know, acquire language in the first few years of life. In Down syndrome, you kind of quickly reach a plateau where you kind of just slowly make improvements over time or not at all. Now, not all aspects of brain function are affected in individuals with Down syndrome. Speech and language skills are particularly affected, and this is primarily because of the inability of their brains to encode short and long-term memories. And you can imagine that would have a grave consequence. If you can't learn today what CAT means as a cat, you can't make that association, you can't build vocabulary, you can't build comprehension, and therefore your language suffers from it. Unfortunately, all individuals with Down syndrome by their 30s and 40s will show all the pathology of Alzheimer's disease. And as you know, this is a devastating disorder where neurons are being destroyed in the brain. And so in addition to whatever impairment they have, they also have this impairment, which is going to cause profound effects later. Now, until about the 70s, the standard of care was very, very rudimentary. Around that time, through better nutrition and better medical care and taking the children out of institution, their average life expectancy went from 25 years of age to 45 to 60. So that was a big change for them in their lives. Importantly, by inclusion education and socialization, there was a major change in their functionality. They went from being kind of not even part of our world to being amazingly integrated. That was a big major change in their lives by being able to be part of society, being included in high schools, etc. Unfortunately, there are no formal treatments for their cognitive impairment. This leaves the patients dependent you know, on society for years, long after their parents have gone. Obviously, if you're having a child with Down syndrome in your 30s and 40s, that they will outlive you, especially now with better treatment. So the economic impact alone is enormous. In terms of kind of treatments that have been considered, one of the earliest ones in the 60s, just simply, they thought this was a problem of vitamin and micronutrients, especially from the work of Linnaeus Pauling and others, which said, okay, vitamin C can cure everything. There was an effort to use vitamin supplements in this patient population, but eventual clinical trials proved that while they are important, they didn't change anything cognitively. There are also a variety of others. I'll only mention a couple of them. Because of the connection to Alzheimer's, there was a thought that the drugs that were being developed for Alzheimer's patients, in particular those that were affecting the levels of acetylcholine, which is a neurotransmitter that's important for kind of awareness and attention, that they tried a variety of these drugs in these patients and they had no effect at all. So effectively, as of about 10 years ago, which is relatively recent, there were no known drugs shown to be effective for improving any aspect of brain function in Down syndrome. So there's been effectively, in the last 10 years, a revolution in the neurosciences with the recognition that many of these disorders, in autism, Down syndrome, you know, that these are actually addressable pharmacologically. To actually develop therapies that can address either behavior beyond just simply let's try drug X, Y, and Z, which is how psychiatry was driven until the 70s, where we would just experiment on you willy-nilly. I would say, let's just try drug X, Y, or Z on you, and we'll see what happens. So this was happening in institutions across this country and around the world extensively. So a lot of the drugs, like we know, lithium, etc., were used because they seemed to do something in this population versus another. We were experimenting on humans. So this is no longer allowed. You're not allowed to do this, although doctors can do a lot. So the strategies that modern neuroscience are taking is first to say, well, what is wrong in in patients with any given disability, like Down syndrome. We largely are using animal models, a variety of different ones, that we hope will reflect the human condition. And in so doing, we'll be able to define mechanisms and then develop rational therapies. So this has been like a wave that's passing through our community, and the impact of this is huge. So what is Down syndrome? I already mentioned that. These are all the chromosomes in human. And it turns out that in Down syndrome, chromosome 21 is found in 3 copies. So 95% of all patients with Down syndrome have 3 copies of chromosome 21. And about 5% have what we call partial trisomies, where a portion of chromosome 21 will be tethered to another chromosome and propagated through the individual, affecting their many facets of their brain and morphological features. So what does it mean to have an extra chromosome? Well, you all know what chromosomes are. The chromosomes are found in cells. They actually are composed of DNA, and DNA encode genes, and these in turn encode proteins. So DNA gives rise to mRNA, messenger RNA, which then gives rise to protein, right? And so all of our cellular functions are defined by the proteins that we have. So we can take a piece of DNA and it makes RNA. We're just representing the exons of that gene in different colors. We combine them together, and this then gives rise to a protein which has several different parts and domains. Now, how do genes affect who we are? Well, clearly what kinds of genes that we have affect whether we're, for example, short or tall. And this is really a balance of, like, growth hormone will be an interesting example of a protein which could do this. And then if you have a certain amount of that protein, you can either be tall or short. And the amounts of those proteins really affect what you become. And if you have more of those proteins, for example, more growth hormone, we can make you be larger. Of course, if you eat a lot of McDonald's hamburgers, you don't need growth hormone. You can also, from the environment, become very large. And my son, who's 6'4", is a good example of that. Now, I won't say it's due to hamburgers, but maybe some hormone steroid that's in our beef. Okay, and so then you get someone who's much bigger. So in the case of Down syndrome, what we have to face is that there are 300 of these genes, which are triplicated. So this is a classic gene dosage disorder. It's a disorder in which we have one extra copy of a gene, or more than one. This means that you have 50% more of that protein, and it's affecting the equilibrium, which is driving you to have some new characteristic. It doesn't necessarily mean that, so you get new characteristics. So in the case of Down syndrome, one could predict that there are some of those genes that are responsible for the craniofacial features, the length of the bones that they have, their stature. They're largely shorter. They are often overweight. They eat too much. They of course have all these cognitive disorders. So that's somehow all of those genes, or some subsets of those genes, are actually responsible for the phenotypes that we see. Okay, this is clear. These are kind of simple concepts. I just want to make sure we're all on the same page as we think about that. So if we now kind of walk through trying to define what's wrong, I told you that one thing that's wrong is we have all these extra genes. But how do these genes play out in terms of how the brain is working, and what parts of the brain are working or not working? So really elegant work that's been done by Lynn Nadell and published in the early part of 2003 basically has characterized some of the main features, some of which I've already mentioned to you. Learning is normal in very, very young subjects, less than 6 months of age, but declines progressively in the first year. This decline occurs in adults with a risk of early onset Alzheimer's making a further impact on them. And I'll just drop down to what's really intriguing is that impairment is not across all learning and memory systems. Now we think of learning and memory as a form of I experience this room and the people that are here and this talk today. This is then logged into short-term memory. And then during your sleep, oddly enough, this is a time in where those short-term memories are moved into long-term memories through your cortex, which you can then have access to tomorrow as a result of your sleep, and the next day and years to come. Okay, so there's a kind of two-step process of that. And the hippocampus is a part of the brain. This is absolutely essential for short-term memory acquisition. Our sensory information comes in, it's processed by our hippocampus, and this allows us to then encode information. And then it's the movement of that information into the cortex which gives you your long-term memories. And these are really disrupted in this patient population of all the memories. So their cerebellar memory functions which are involved in movement and gait are not impaired. So it's not like all systems are affected equally. And as I mentioned earlier, these have really big effects on language, speech, and verbal. So if we want to figure out more about what centers of the brain are doing this, we can look at issues of neuropsychology, but we can look at anatomy. And if we look at the anatomy of the brain of an individual with Down syndrome versus a healthy individual that doesn't have 3 copies, what we notice is that the cerebellum is much smaller. You can kind of see that the whole cortical areas are actually reduced, the number of gyri are reduced, and in fact the hippocampus, which I just mentioned, is important for memory function, is smaller. Further studies have looked at the anatomy. If you just take a segment of the cerebral cortex, each one of these dots represent an individual cell, neuron or glia that's in the brain, and you can see just by comparing an age-matched control that the number of cells that are making up the cortex are a lot less. So this implies that there's some deficit during brain development which is affecting cell number. Once those cells migrate into the right layer, so this is the parietal cortex of a newborn, and you can see where all these individual neurons with their dendrites or axons are making up layers that make up the cortex. Within the first year of life, 2 years of life, the total number of cells don't change, but what does change is the arterization of these cells. And as I'll show you in a second, this arterization is related to the number of synaptic inputs that a cell has. Those synaptic inputs, synapses are, and you'll see in a second, are the seed of learning and memory. So how complex these arbors are tells you something about how well the brain is able to learn. And in Down syndrome, you have more of this situation than this one, indicating there must be some problem with the wiring and the number of connections. A very cool way to look at this is to look at one cell. And here is one cell, one neuron, in a cat visual cortex, which has been filled. And you can see where its entire dendritic arbor is quite extensive. This is the soma. These are the dendrites which receive all information. And then the axon, which is a single axon, which comes out and then goes and makes contact with other cells, is quite elaborate. This has actually been squashed into 2 dimensions, but in fact it's like a Christmas tree is in 3 dimensions and covers a lot of space. So if you jump back and place this one cell into this cortical layer, each one of these spots represents one of these really incredible dendritic trees that are woven in between each other. Okay? Now, what happens is if you just focus on one dendritic segment and blow it up, this is one dendrite here, you'll see that the dendrites have these things called thorns or dendritic spines on the surface of them. And if you look at these thorns by electron microscopy, this is one of them here, you'll see that each one of them is contacted with a presynaptic bouton from an axon, from another cell, okay? And so this unit is called the synapse, and it works by taking neurotransmitters which are stored in these vesicles. They go and fuse with the membrane, they release the transmitters, they act on a receptor on the postsynaptic side and trigger an electrical signal that goes to the brain. Importantly, this little unit here is what encodes every single bit of information that we take in. It takes about 1 million neurons and about 3 synapses per connection to encode one memory trace, which would be your first glimpse of the mountains showing some horizon. The next memory trace would be your car driving into the campgrounds at Yosemite or Tetons, etc. And so those memory traces add one to the next, and it takes about that many neurons and that many of these synapses to encode one bit of information. Okay? You can interrupt me anytime. So what's important about that piece of information is that that actual number of these spines tells you how many synaptic contacts a cell is able to make. And in this particular case, most cells make 20,000 synaptic contacts. So we can play a very interesting, we can perform a set of experiments in Down syndrome and look to see whether these spines are forming in normal numbers over time. And so this is normal development, where a dendrite has very few synapses to begin with and very few spines. And about by 1 year to 2 years of age, you can see that the number is enormous. Furthermore, they kind of become from long and thin to being very regular in shape over time. So these are hallmarks that there's synapses. And if you look at an individual with Down syndrome, these spines are long and thin, and some of them are big, and their numbers are really small. So these are all elements that argue that there's a synaptic defect. Okay? So that gives you some of the background of Down syndrome, that we think that there are deficits in synaptic learning, that they're affecting the hippocampus and certain parts of the cortex, and as so that gives us a structure to begin thinking about what we can do. However, as I said, since the 50s, we aren't allowed to experiment on humans, and you probably appreciate that I don't come asking for you to give me some of your brain tissue that I can experiment on. Of course, we can use fMRI and other things like that are non-invasive, but those techniques don't give me single-cell resolution. They really don't tell me how the circuit is functioning. So there are a variety of animal models. This one here is a C. elegans, which is about 1 millimeter in size. This is a Drosophila. Both of these systems are great for genetics. But almost all studies that are looking at behavior-related things and genes and trying to copy human condition, mostly use mice or other rodents. Higher animals are almost not used, although there are some that are using higher level organisms. But for all the studies that I'm going to tell you about relate to the mouse. If we wanted to create an animal model of Down syndrome in the mouse, we have to consider that we want to basically create a trisomy. We want to be able to take the genes that are found on chromosome 21 and make 3 copies of them in a mouse. So human chromosome 21, I told you, has 300 genes. And in a mouse, those 300 genes are actually placed onto 3 chromosomes in close blocks. One is about 200 genes here on chromosome 16, 50 more on 10, and 50 more on chromosome 17. So if you wanted to make a trisomic mouse, you would have to take this block of genes here, here, and here, and triplicate them. Now, using chromosomal engineering, this has been accomplished to its entirety within the last year by Eugene Yu. So that mouse has been created. But there have been other mice that have been existing for some time which only take this distal end or this segment of chromosome, chromosome 16 and triplicates that. And that's shown here. So that all the work that I'll just mention to you has been done with this one mouse. And what's really interesting is that this mouse has like 80% of all the phenotypes seen in Down syndrome. So for example, I mentioned to you about the cerebellum being smaller, and in this mouse model, the cerebellum of these mice is smaller. The hippocampus is smaller, the cortex is smaller. They have the same craniofacial deficits. They have the same learning deficits, which are hippocampal and prefrontal base. So this argues on many levels that these are pretty good, not perfect models. They don't have heart defects. They don't have leukemias. They don't have all the features, but those must be present on some of these other chromosomes. So to explore using these animal models what's wrong, what we use is something called physiology. This basically puts an electrode into a cell and listens to it, and this is a cell here firing. Kind of like Morse code, okay? And we can listen to them, and we can learn a lot about them. So the area of the brain that we're most interested in is the hippocampus, which is shown here. And most of that came from a gentleman named Henry, H.M., he was known for many years, he just passed away recently, who had damage to his hippocampus. And what's interesting is he had only long-term memories, what he had learned before, and he had no short-term memories. He could see you, and you'd introduce him. Hello, Albert. My name is Craig Garner. And Albert, if he was HM, would say, hi, Craig. And then 5 minutes later, I'd walk over here, and I'd come back, and I would say hello to him again, and he couldn't remember me. Okay? And in fact, this happened when he was 21 years of age. And when he was 50, if I showed Albert, HM, a mirror, and he looked at himself, he would go, who is that? Because he had no way to store long-term memories. So the hippocampus is a really important part of our brain for this. So we can go into the hippocampus, we can use electrophysiology, and we can listen to cells as we give them a stimulus and ask for their responsiveness. And the result of this has told us some very interesting things about what's wrong. First of all, what we've learned is that the synapses are able to encode information, but one set of synapses, which are illustrated here, which actually drive inhibitory tone, are much too active. So if we just look at the downward spikes here, this represents individual synapses firing of the inhibitory type, and you can see that the numbers are much increased in these brains. So what this tells us is that, and because of this, these synapses are unable to learn. So an example of learning is I give it a pulse, it responds of electrical pulse, and it responds with a postsynaptic response. And I do this several times. And then I give it a tetanus and say, okay, I want you to really learn something. And most synapses get stronger and they'll hold that new strength for some time. Whereas these animals are unable to do this. Now the reason for this is that, that the inhibitory tone in these animals seems to be too high. And this is suppressing synaptic plasticity and thus interfering with neural circuits. Okay, so by studying these animals, we've learned this. And in doing so, we're able to actually make a prediction about what's wrong and actually then create a strategy for treating this disability. So to do that, our prediction, of course, is there's too much inhibition, and we want to use drugs that will reduce that inhibition. And we developed a model that actually can test this in our animals. It's based on object recognition. And children with Down syndrome cannot do this. This basically, I give them 2 objects today. And tomorrow, I'll give one of those same objects again, plus one new object. And invariably, they will not be able to remember that they saw that other object from yesterday. And part of that is measured by attention. So what we do when we see a new object is we spend more time exploring it, ignoring the other. Okay, and so the degree of intellectual disability correlates with your error rate. So this can be actually done in these mice, and this is just a movie showing mice that are in 4 different chambers. These are objects, and they spend a certain amount of time going up and touching them. And if it's a novel object, they'll spend a lot more time with it than another. So doing this type of experiment, we can actually examine whether these mice have deficits in learning. So in these experiments, we're actually today training them with 2 different objects. The next day, we give them one of the same and one new object, and we have a measure of the index of discrimination. And a normal animal will actually spend almost all this time with the new object, and it turns out that this mouse model spends equal time with both objects, indicating that it hasn't learned that problem. So Fabian Fernandez, a graduate student, a really talented graduate student in the lab, came up with this idea, and what he then did was take a simple drug, pentylene tetrazole, which can be used at very, very low doses to remove the GABA inhibitory tone, and ask whether or not learning would be restored. In this example here, 1 week post-treatment of this drug given in chocolate milk, we just put a little thimble with chocolate milk in it with this little drug. It's like 10 microliters. They eat it today, and then we do our testing, etc. So what happened is after we gave it for 3 weeks is that you can see that the performance of these animals on pentylene tetrazole is the same as wild type. Really, really coolly is that 2 months after they've had a 3-week regimen of this drug, they are performing still as well as their wild-type littermates. This was really profound. This was really, really cool. This was really exciting and suggested many things. It said for the first time that intellectual disabilities in Down syndrome are A, caused by too much inhibition, and secondly, that it could be treatable by giving low doses of a drug. So this is very cool. We published this in 2007, and since then, our group has founded a company to develop this drug. We've actually done all preclinical studies to show it's safe, and we are entering a phase 2 clinical trial in Down syndrome in this spring. So March of 2012, we began our study, began evaluating this drug.
So what exactly is the drug doing again? It's preventing the inhibitor.
It basically reduces very, very slightly the amount of inhibition. And what happens over time is that if you give it once a day, your inhibitions is excitation inhibition, so excitation is where we're learning. Inhibition, if it's too high, you can never learn. And normally what happens in our brain is that this is in this state, and when some new information comes, inhibition is turned off, we learn something, and then inhibition comes back up, and we keep that memory intact. And what's happening here is the inhibition is so high that they never learn. And the idea was if we give a little bit of this drug every day, that you will slowly drive down the inhibition because the excitation gets stronger, and that balance will come back at equilibrium. Okay? Yes?
So, is it like an anti-function of a depressant medicine?
So it's similar in many regards. All drugs, and I'll mention just a few of them and then stop, is that drugs that we use in neuroscience are all acting on neurotransmitter systems. We have inhibitory systems like GABA. GABA, of course, has been the favorite target of Roche and other major companies who make Valium, and drugs that inhibit or increase the activity of the GABA receptor, causing you to go to sleep or sedate you. SSRIs are drugs that actually act on the serotonin system, another transmitter, and affect depression. And other ADHD drugs do something similar. So conceptually, you realize now that we're almost there, right? We're actually at the door of being able to treat patients with Down syndrome for their intellectual disabilities. And the drug that I mentioned is phenyltetrazole, and it's acting to normalize excitation inhibition. But we're not alone. Since we published this paper and said this was possible, many, many other groups have looked at all these other systems. So there is work done with fluoxetine, also known as Prozac, an SSRI, which can be used to normalize the number of cells that are being born in your brain. Notably, our studies and theirs were being done in adult individuals. So what that means that you can actually intervene in an adult, you don't, or a child, you don't have to intervene at birth. But there are some that you might have to do that with. There are even others like Focalin, which is an ADHD drug, and these mice, as kids with Down syndrome, have ADHD. So what you're really facing in terms of developing a treatment that will remove as many of these phenotypes as possible is some cocktail of drugs that are ultimately going to be necessary.